Hello, everyone, and thank you for joining us today for this joint webinar, which is the 11th on the use of NOMS and risk assessment, and the 6th in the neurodynamic discussion series. This webinar is being co-organized by the Physicians Committee for Responsible Medicine, the U.S. Environmental Protection Agency, the PETA Science Consortium International, and Unilever. I'm going to give a second just to let more people log on. We had quite a few people register for this webinar, which was great. But I'll go ahead and introduce myself. I'm Bridget Rogers. I'm an advisor to the PETA Science Consortium International, and I'll be co-moderating today's webinar along with Gino Scarano, who is the Senior Advisor in the New Chemicals Division from the EPA Office of Pollution Prevention and Toxics, as well as with Christy Sullivan and Phoebe Woodruff, who are with the Physicians Committee for Responsible Medicine. Okay, I'm glad y'all are saying where you're from. Thanks for adding it in the chat. Before I introduce our first speaker, I'd like to point out that the slides and a recording from today's webinar are going to be available at the links on the screen, and the slides and recording of the first 10 webinars in the NOM series and the first 5 webinars in the Dynamic Discussion series are also available at those respective links. Today's webinar is going to be part 1 of a 2-part series, and part 2 is going to be held on January 18th of next year. The registration for part 2 is actually now open, so you can scan the QR code that's on the screen now, or follow the link in the chat below to register for that second webinar. We have 2 speakers today who will be presenting on frameworks for establishing scientific confidence in new approach methodologies. We will then go into a panel discussion with both our speakers as well as 2 additional panelists. We'll take questions that you can enter into the Q&A box at any time. Please write down if they are for a specific speaker or a panel member, okay? And I'll go ahead and turn it over now to Christy to give us some updates. Christy?
Hello, everyone, and thanks, Bridget and Phoebe, for starting us off. So as Bridget mentioned, this webinar is a partnership between 2 ongoing webinar series because we realized that we were trying to close out what has been a very eventful 2022 with 2 very similar events. Today's topic, I think, is on everyone's mind. I don't think that's an exaggeration. And so hope you will enjoy participating in today's discussion and mark your calendars for part 2. And we're also hard at work planning the dynamic discussions events for 2023 starting in February. So keep an eye out for announcements or join our mailing list.
Okay, thanks, Christy. I'll now turn it over to Gino Scarano to give some comments. Gino?
Thank you, Bridget and Christy. Hello, everyone. It's my pleasure to welcome you, the speakers, the panelists, and all of you that have registered to join today to discuss this important topic. I mean, in my view, the identification and use of new approach methods, or NAMs, by regulatory agencies in the US and around the world is a journey and a goal that has been around for at least a decade. The framework and approach that's going to be presented today, I think, will help us on this journey. And I'm looking forward to the presentations, the discussion by the panelists, and questions from all of you who have registered. So looking forward to it. Thank you. And let's go get going.
Great. Thanks, Gino. Okay, well, we'll get started. Speaking first today will be Dr. Nicole Kleinstreuer. She leads NICEATM, the U.S. federal resource for alternatives to animal testing. NICEATM's work is centered on domestic and international efforts to develop novel testing and analysis strategies that provide more rapid, mechanistic, and human-relevant predictions of potential environmental chemical hazards. She has a secondary appointment in the NIEHS Division of Intramural Research Biostatistics and Computational Biology branch, as well as adjunct faculty positions at the Yale University School of Public Health and the Eshelman School of Pharmacy at the University of North Carolina at Chapel Hill. Welcome, Nicole. Thank you so much for being here today. Please go ahead and share your slides and get started.
Great. Thank you so much, Bridget, and thank you to all the other organizers. This is really such a fabulous webinar series. I'm really privileged to be presenting in it today. And I personally have learned a lot from all of the previous webinars in this series. So it's really exciting to be part of it. Okay, so today I'm going to be talking about kind of the evolution of validation, both from a US perspective and then how that fits into global efforts as well. So first, I'm going to start with a document that I hope you are all very familiar with. If you're not, the easiest way to find it is to just Google ECVAM roadmap. And this is something that was published almost 5 years ago now, but I think is really still extremely relevant and extremely current. And it is the US strategic roadmap for establishing new approaches to evaluate the safety of chemicals and medical products. And it's I think, fundamental concepts within the roadmap are really exemplified by the figure on the front cover. So we have this iterative approach which encourages constant communication between end users, method developers, and bodies like NICEATM, ECVAM, ECVAM, other organizations that are trying to validate or establish confidence in these new approaches such that they are fit for regulatory use, such that they are fit for the purpose that they're intended for, and therefore will ultimately be adopted by our regulatory decision-making friends and by regulated industries. But I think one thing that really came out during the development of the roadmap and has just continued to be increasingly apparent is that there is really no such thing as a one-size-fits-all approach to gaining regulatory acceptance of a new approach methodology or a NAM. So I think the erroneous perception previously was that validation was just a checkbox. So you have a new method, you ask for it to be validated, so you run a validation study, and then once it is validated, then you've got that seal of approval and you have just blanket regulatory acceptance. And really that could not be farther from the truth. So just as an example of two different US federal agencies that engage in regulatory decision-making, the FDA and the EPA, even within those two agencies, there are many different centers and offices that deal with different regions of the chemical universe and have different contexts of use, different purposes, different regulatory decision frameworks. And for a method to be validated for those different contexts of use, it needs to consider those purposes and those decision frameworks up front. And a level of confidence has to be achieved that is appropriate and tailored to those particular contexts. And that includes things like biologics or medical devices, food additives, cosmetic ingredients, pesticides, different industrial chemicals. All of these different types of chemistries and different regions of the chemical universe and different exposure scenarios have different considerations when it comes to regulatory decision making. And so for a method to be useful in those regulatory decision-making frameworks, all of those considerations have to be brought in at the beginning of the conversation as the method is being developed. And then the process of validating or establishing confidence in that method has to be iterative, where the regulatory decision makers and their context of use and their decision frameworks are constantly being considered at every step of that process. So what we're looking at here as we're evolving the concept of validation is going from this kind of one-size-fits-all, yes, no, something is validated or not for, standalone methods as replacements, going from that sort of outdated concept to more of a decentralized approach where the end users are extremely engaged in the process. The process of establishing confidence in a method is not only flexible and fit for purpose, but it's an evolutionary process. So you may establish confidence in a method for a particular purpose, and then as that method evolves, you may establish more confidence in it for a wider variety of purposes. And ECVAM, which is a committee of 17 different US federal agencies has been working very hard on updating their guidance around validation of new approach methodologies. So the old ECVAM guidance is from 1997. And so we have decided to basically completely overhaul it, and we have a validation work group that is working on a white paper that kind of encompasses all of the concepts that I'll be talking to you about today. And so this is a sneak preview from that white paper. This is one of the main figures, and it borrows a lot from some existing publications that I'll talk through today. But some of the unique aspects of the validation guidance are things like the context of use terminology. So emphasizing that processes to establish confidence should be flexible, adaptable, and fit for a particular purpose or a particular context of use. Emphasizing the need for communication because regulatory needs vary across agencies. And then a number of essential concepts around things like biological relevance, technical characterization, data integrity, and information accessibility and transparency that I'll talk through in more detail. So those concepts are extremely consistent with a number of existing publications in the literature. And I put these 3 publications in the order that they're in on the slide very intentionally, because this publication, the van der Zalm paper, very recent, just came out in the last couple months, is the one that I'll be focusing on, but it really rests very firmly on the shoulders of these 2 pre-existing publications. So this OECD guidance document 34 it came out in 2005, and it is certainly, I think, an evergreen document. It maybe could use some refreshing and some updating, but all the fundamental principles of GD34, I think, still hold true today. And then in the context of developing defined approaches for skin sensitization, there was an international publication that came out of NICEATM, the International Cooperation on Alternative Test Methods, that laid out an evaluation framework for defined approaches that, again, have very consistent concepts with the scientific confidence framework that is detailed in the van der Zalm paper. This slide also serves as my acknowledgement slide because all of the co-authors on the van der Zalm paper are the folks that I need to acknowledge for their very substantive contributions and their leadership in this space. So diving into detail in terms of what a scientific confidence framework looks like for establishing confidence in NAMs, this is the key figure from the paper. So you can see the 5 main elements here, which are all interconnected. Are fitness for purpose, human biological relevance, technical characterization, independent review, and data integrity and transparency. And that interconnectedness is important because the order of when you might look at these elements can vary, and some of these are done iteratively or in parallel. And so I'm going to go through kind of each of these elements and some examples of how they're being considered in real world case studies. And then TAala Henry, the next presenter, is going to, I think, dive a little deeper into specifically what the EPA is doing in terms of applying a scientific confidence framework like this one to methods that they may use in their regulatory decision contexts. So beginning with fitness for purpose, this schematic shows some of the questions that are relevant to determining fitness for purpose of a NAM. And I want to draw your attention to the synonymity between the phrase context of use and purpose. So with the ECVAM validation guidance, we're using the term context of use, but that's really the same as the purpose of the method. And so what are some questions that you might ask yourself are, what regulatory statutes the data from the NAM are intended to comply with, and ensuring that the NAM provides information that is really sufficient to address those regulatory statutes and the endpoints of interest, and will allow end users to actually be able to come to a conclusion for the chemical under consideration. So are these, context of use things like classification and labeling, or deriving a point of departure for quantitative risk assessment, or obtaining additional mechanistic information for a weight of evidence analysis, etc. So these are the concepts of fitness for purpose. Understanding human biological relevance is, I think, something else that's really key in terms of this evolution of establishing confidence in a method. And I have human in parentheses here because in the Van der Zalm paper, we really focused the application of the scientific confidence framework to predicting human health endpoints. But in the ECVAM validation guidance, we're expanding that because we understand that environmental safety and ecotoxicology predictions are also extremely important. And so biological relevance is not only important with respect to human endpoints, but it's also important in terms of other species of relevance or other species of concern as well. And so the concepts here are looking at the similarities between the physiology of the test system and the biology measured by the test system and the biology of the species of concern. And in this case, we're looking at humans. And so the concept is that confidence in a NAM is really increased when it adequately reflects human biological understanding. So that, for example, could be mapping to a key event in a relevant adverse outcome pathway, but not necessarily. And I think, it's always nice to be able to compare the output of a NAM to human data where it's available. Oftentimes it's not going to be available. And so when you're confined to comparing NAM outputs to things like traditional animal test methods, it's also important to take into account the human biological relevance of the reference data itself. And so when such comparisons are appropriate, demonstrating that the NAM reflects human biological understanding as well or better than the traditional animal test method can also help increase confidence. And so I think a really nice example in the literature of how that's been done for a particular endpoint is for eye corrosion and irritation potential. So this paper was published last year, and kind of the motivation for looking at this in a different way was a previous analysis of the ECHA database that looked at about 500 substances with repeat animal tests. So at least 2 Drey's rabbit eye tests. And when you look at the reproducibility of those independently run animal studies on the same chemicals, it's pretty abysmal, particularly in those middle categories of mild and moderate irritants. In fact, a mild or a moderate irritant from a first test is actually more likely to be classified as a non-irritant the second time it's run. And so that demonstrates that the rabbit test itself is really not a good benchmark and probably shouldn't be used as a reference method to demonstrate the validity of new test methods that are trying to predict this human health endpoint. And so in thinking differently about how to show the validity of new methods, we focused here on human biology rather than a direct correlation to the rabbit test. And so looking at, for example, the depth of injury model, which basically says that the depth of injury in the eye correlates with the severity of injury, and then considering the strengths and limitations of all the available methods, in vitro and in vivo, with respect to their relevance to human ocular anatomy and physiology and mechanisms of eye irritation and corrosion in humans. I think another very well-known example of this is the defined approach for skin sensitization work. So here we have a well-established, vetted adverse outcome pathway for the mechanism of how allergic contact dermatitis or skin sensitization occurs in humans. And this recently adopted internationally harmonized guideline from the OECD shows that when you have testing strategies that are comprised of NAMs that are mapped to key events within an AOP, so based on human mechanistic understanding, they actually outperform the animal test when compared to human reference data, because this is a unique scenario where we actually had a highly curated, robust and reliable set of human reference data. And so they outperform the animal test when compared to that human reference data. And they are equivalent to the reproducibility of the animal data. You know, if you played the thought experiment, you know, what would happen if we didn't have the human reference data, which is the case for pretty much every other endpoint. So the next aspect to consider in the scientific confidence framework is the technical characterization of the assay. Of course, this is something that's going to be really important for any scientific method. So evaluating the protocol, the equipment used, any models being used for endpoint prediction, assessing and understanding the reproducibility, the transferability, if that's applicable, the applicability domain, any technical limitations of the method, how, you know, what are the associated reference chemicals and controls, what are the limits of detection, and having a data reporting and a method reporting structure that allows for a detailed and transparent assessment of all of those aspects. And, you know, obviously, part of technical characterization of a method is assessing the performance of that method. And that is generally done via looking at accuracy. And so accuracy typically relies on a comparison to data from the reference method, which is usually the traditional animal test method, right? But I think it's important to evolve our thinking around that. So while that is historically the way that results from a new method are assessed, so comparing to results from the traditional animal test method, it shouldn't necessarily be the default way to determine the accuracy and relevance of a method. We know that traditional animal test methods themselves are surrogates for the endpoint that we're trying to predict, which is perturbation of human biology and human toxicity outcomes. And so assessing the benchmark method, the animal reference data, in the same way that you would assess the new method is critical. So looking at the relevance of the animal method to the human health endpoints under consideration, understanding the variability of the animal data, and using that to set appropriate expectations for quantitative performance of a new approach that's being used to predict that same endpoint. And kind of moving past, you know, a exclusive reliance on comparing a new approach to the outcome of the existing reference animal standard, and incorporating additional considerations like how consistent are the results across different methods that are intended to predict the same type of endpoint or measure the same biology. A greater reliance on reference chemicals where we have a high degree of confidence in their activity or inactivity. And again, a much greater emphasis on biological relevance and reproducibility in the context of accuracy. And there recently have been a ton of analyses that have tried to start to provide some of those resources to understand the variability of the animal reference standards so that we can create more appropriate expectations and quantitative benchmarks for performance of new approach methodologies that are intended to either supplement or fully replace some of these reference animal standards. And one example where we've tried to incorporate the variability of the reference data not only in evaluating the performance of a new approach, but also in applying data-driven confidence intervals to model predictions is the Catmos project. So Catmos is the collaborative acute toxicity modeling suite. So it is actually a crowdsourced compendium of models that are intended to predict the acute oral systemic toxicity of a chemical. And so we built consensus models from 135 different models submitted to us from groups around the world. And so we have really robust models that cover the entire chemical universe with defined structure. And we have, in conjunction with that, we curated quite a lot of data from the existing rodent oral LD50 test. And in doing so, we had several thousand chemicals that had independently replicated studies. So we were able to take a data-driven and quantitative approach, bootstrapping through the standard deviations from those replicate animal data to actually determine how variable it is and derive a confidence interval, a 95% confidence interval that can both be used to help assess the performance of the model, but also to put confidence intervals around the model predictions. And so we're partnering with the EPA right now to look retrospectively at risk assessments and see whether or not we could replace the in vivo LD50 data with the in silico, computationally predicted LD50 data and arrive at the same risk conclusions and regulatory decisions. There's also additional guidance that's been published recently on how to really apply things like quality control and other sort of statistical data analysis and reporting and evaluation, conceptual evaluation approaches to really build robust and reliable NAMs. And so this is a figure from a paper that was led by NIST and several other ECVAM colleagues, talking about a technical framework for developing robust NAMs, and having consideration among all of these aspects will, I think, help bolster confidence within that scientific confidence framework. And then the last two elements that I want to talk about are data integrity and transparency, and then how that feeds into the ability for independent third parties to review all of the information around NAMs. So having data integrity and transparency and demonstrating the credibility of the data submitted for assessments and peer review is obviously incredibly important. In terms of increasing confidence and being able to assess and describe the uncertainties and limitations of any particular method. And that should all sort of feed into the ability to have an external review around a NAM. And so that could be done potentially through a peer review process, publications in peer review journals, ideally having some sort of independent scientific advisory panel review a NAM. We're trying to move away from this centralized approach where that's simply the purview of the validation bodies, but also incorporates a lot of interaction and communication with method developers, but acknowledging the importance of having someone external to the lab or the company that developed the method, manage that peer review as well. And so having that independent third party assessment, however that comes about, and we don't want to be too prescriptive around that, but I think it's really important in terms of increasing that confidence and demonstrating the robustness and the reliability of the method. And there's a number of documents and guidance that are out there on standards for demonstrating reliability of a method, standards for data integrity. So this is the OECD guidance document on good in vitro methods practices, which is really focused around sort of protocol aspects and how to build robust NAMs. There's also policies that have been published by the OECD on confidential information in test guidelines and how that can be made available to independent reviewers and in particular national coordinators of the test guidelines process to help facilitate review of the reliability and the performance of that method during the development process, and then evolving towards making that types of information more publicly available. And then there are also a number of resources online that try to transparently communicate and, as far as possible, make information on the various methods publicly available, both in terms of how what the methods consist of, but where they are in the validation process or in the process of establishing confidence in them. And the two that I've highlighted here are the TSAR, the Tracking System for Alternative Methods Toward Regulatory Acceptance, which is a really phenomenal resource that the JRC part of EURL ECVAM hosts on their website. And then also on the NICEATM website, we have a list. It's over 125 entries long now of alternative methods that are accepted by US regulatory agencies, and background on the method and the sort of applicable regulation and regulatory framework that they fall under. So in summary, I think, hopefully I've communicated to you sort of the evolving concepts around establishing scientific confidence in new approach methodologies that are fit for purpose, that consider the context of use in regulatory decision-making, and how the different elements of that framework can help increase confidence and translate into adoption and implementation of these models. So I will stop there and I will stop sharing.
Great. Thank you so much, Nicole. That was a really excellent presentation. As a reminder to everyone, we're going to hold all questions until the end of the second presentation, but feel free to submit them now in the Q&A panel. I see we already have a few coming in, which is great. Okay, so we'll move on to our next speaker now. Speaking next will be Dr. Tala Henry, who is the Deputy Director of the Office of Pollution Prevention and Toxics at the U.S. Environmental Protection Agency, where she leads the assessment and management of new and existing chemicals under the Toxic Substances Control Act. She has long championed computational and other new approach methods for application and risk assessment. She has recently co-authored 2 publications related to NIEHS, one on the framework for establishing scientific confidence in NIEHS that we're discussing today, and another on the use of NIEHS to meet regulatory requirements for chemical assessment. Tala, thank you so much for being here today. Please go ahead and share your slides and get started.
Okay, I think I shared. Do you see them?
Yes, go ahead.
Okay. Again, thanks to the organizers for the invite. It's a pleasure to be here and keep talking about progress being made in applying NIEHS within the regulatory framework. So first, I'd like to acknowledge in particular Allison Harrell from our Office of Research and Development here at EPA, and Anna Lowett in the same Office of Pollution Prevention and Toxics as I come from, because all these slides are theirs. So what I'm going to do here is start with kind of outlining what current work is going on at EPA to develop sort of a formalized competence framework. You're going to hear a lot of the same words and concepts that Nicole just covered, but we're putting that within a framework that's fit for purpose for EPA application of NIEHS. And then I'm going to move into some specific examples of how EPA programs, particularly the chemical assessment programs, the Pesticides Program and the Toxic Substances Program, have been making progress in taking up NIEHS within that regulatory context. So fit for purpose there. So first, just want to rewind a little bit. In 2020 and then updated in 2021, and the updates are on the second bullet here. You can read those yourselves. But the Office of Research and Development and the Office of Chemical Safety and Pollution Prevention here at EPA were the leads in developing a new approach methods work plan. This was how EPA specifically, it is an EPA wide document. The 2 lead offices probably do conduct or require the most toxicity testing across the whole agency. That's why we were the leads, but nonetheless, it is a work plan that involved all different EPA program offices. And you can see of the 5 objectives for achieving the reduction goals put forth in this work plan, one of which is to establish a scientific competence and demonstrate its application. So again, I'm going to talk about both of those things here today, starting with this competence framework. So the goal of the framework that is under development now, and if you were at our EPA NIEHS conference, you may have seen some of these upcoming slides, but nonetheless, our goal here is to develop a more generalizable scientific competence framework that's applicable across a broad range of NAMs, as well as across EPA agency decision contexts. So again, it is, the work plan is an agency-wide thing. Certainly pesticides and toxics programs, again, either, and the Office of Research and Development conduct or require the most testing, but nonetheless, the whole agency can use this framework going forward. So the initial framing of this competence framework is based on many of the things you just heard Nicole present on. So it has a really strong foundation in many different scientific resources that have been published in the past and are emerging that focus on specific types of NAMs or specific applicability domains regarding toxicity endpoints, for example. So I include here a lot of words, but this is EPA's working definition of what is a NAM. So it's to include any technology, methodology, approach, or combination of such approaches that provides information on chemical hazard and risk assessment, while at the same time avoiding the use of animal testing, because of course reduction in animal testing is the ultimate goal. The use of NAMs allows the EPA to meet these very objectives of reducing the reliance on vertebrate animal tests. And of course, though, we do have to do this in a scientifically justifiable way. And there are probably the strongest and most straightforward now mandates on EPA are through the Toxic Substances Control Act. There's a whole section that was added in 2016 that mandates that our office do certain things with regard to reduction in animal testing, including a strategy I'll get to in a moment. But also, I will say that our pesticides program, which has a long-standing data requirements that have historically been very animal intensive, have been on this journey towards reduction for quite some time, even in the absence of any specific statutory mandate. So in trying to conceptualize what this EPA competence framework should look like, some essential elements have been identified. I'm going to walk through some of these, and as I mentioned, many of these words are similar to things that Nicole just talked about. So there's seems to be a convergence of specific elements that everyone agrees needs to underlie the development, validation, application of NAMs in order to provide that competence. So first of all, we seem to be in agreement that the technology, method, or analysis that is associated with any new approach method needs to be transparently described and of sufficient detail to enable an independent review that Nicole just talked about, and also evaluation of results. Depending on the type of NAM, as we just heard, there is a wide variety of things that fall within that definition I just mentioned of NAMs. So again, one size doesn't fit all, and a particular template may be useful to help think through what you should write down to be transparent, but certainly there's a wide variety of types of NAMs. So we need to make sure that the underlying principles, description of the technology and the method has to be clearly documented and published in public access ways so that everyone can be transparent about what that NAM is. And there are issues arising for privately or commercially developed NAMs that there are certain things being worked out that we do need to have certain things be available to meet this transparency goal. The NAMs, as Nicole mentioned, also need to undergo an appropriate level of independent or external review. Again, that's a key element of raising the confidence in the approach. Peer review and publication of the NAM will also help inform, the relevance. It's fit for purpose in any situation. It may be very suitable in one situation or for one purpose, but not necessarily another. That's why you have to be transparent in describing that context. Reliability, and again, Nicole covered some much more specific examples and areas of this, but being a very important component of any competence framework, the reliability needs to be characterized, described, and transparently communicated to consider whether or not that NAM is suitable for the purpose intended. Certainly, some of the basic science principles apply to NAMS methods as with any other. You need to have appropriate controls, document the purity or stability or solubility of the chemicals within the test system, for example. And of course, domain of applicability is a key element of any method. You have to understand both, in this case, since we're generally talking about testing of chemicals, understand which chemicals the NAM or the method works for and which ones it does not. And certainly, of course, a key characteristic of any NAM is what endpoint you are actually measuring, what kind of data you are basically measuring a response for. So there's a couple of domains sort of crisscrossing the NAMS world, both chemical as well as the endpoint of concern. And then this gets back to the transparency a little bit. We have to make sure that there's public availability of the data to follow the guiding principles in FAIR for scientific data and management and stewardship. And then you heard a lot from Nicole about this purpose angle. So the relevance is another key characteristic of any competence framework. One size does not fit all. And so you have to understand for any given NAM, the relevance for its intended use. And that should then go along with any description and characterization of a new method. Oops, did I do that? Sorry. So anyway, relevance to the endpoint being evaluated. And again, this is a very important thing for some of these new approach methods where we're getting more specific mechanistic understandings, and maybe a couple of NAMS methods might be organized within an adverse outcome pathway, for example. The old way with the animals is you have a whole animal, you dose them and you measure a zillion things. The shift to some of these more efficient and more relevant NAMs might need a little bit more contextualization on how they fit together and can be used together synergistically to interpret the adversity being measured. And uncertainty, like any good science method, is something we need to acknowledge and describe for any method, and where appropriate, applicable uncertainties for the NAM should be presented relative to those uncertainties associated with the standard and traditional approaches. And Nicole did a really nice job of describing that what perhaps historically has been considered a gold standard, many publications are showing may not in fact. And so you really have to think through the uncertainty of the NAM in the context of its purpose, not necessarily against another method that may or may not have its uncertainties described well. Just a kind of a wrap up on where we are at EPA in this competence framework. We hope to get this out into the world in 2024. We have a draft outline now, have been gathering some feedback and really trying to flesh this out. We have a draft framework that will probably go out to some kind of, I don't know right now what the plan is for external inputs. But we do this very often, take comments on things and revise, again, with the goal of trying to get this out in 2024, according to our EPA NAMS work plan. So next, I'm going to move into some examples of activities that EPA programs, largely our pesticides program, but also our TSCA program, have been undertaking to build that confidence, if you will. It's kind of the building confidence by doing. So the first slide here I want to acknowledge is that it takes a village. As you saw from Nicole, as well as this presentation, there are many entities internationally, as well as in the US, most notably a lot of that work that Nicole described happening through or with ECVAM, but also many other entities have been collaborating with the EPA to move forward in building confidence in the use and application of NAMs. So I just want to acknowledge them. Also, just this transparency, it's a key component of a NAM, but also the whole idea of building confidence is where you have to be transparent about what you're doing. And actually education is part of this too. That's what these webinars are all about, is to get the community, the interested community together, discuss things, facilitate greater understanding. And I put up this slide about our EPA NAMS conference. Just as one example, these PEP webinars are another great example of just being transparent about the developments that are happening within the world, because all of NAMS, because that all contributes to the building of confidence. So now I'll switch over and provide kind of a very high level overview of some of the things that here at EPA within our Office of Chemical Safety and Pollution Prevention, which includes the Office of Pesticides Program and the Office of Pollution Prevention and Toxics. So we have our FIFRA pesticide office, and then all what we often refer to as industrial chemicals office, both working really closely together to, apply NAMs in a fit for purpose. So within those very specific regulatory contexts. So first thing I want to mention, I've alluded to the Toxic Substances Control Act had some modifications in 2016, Congress added a whole new section that put us on a path toward reduction of vertebrate animal testing. One of the requirements was to develop a strategic plan, which we put out, Gino can correct me, but I think around 2018. And also another requirement was to provide a list of alternative test methods. And so we've developed a list initially, I believe, in 2018, and we've updated it a couple of times since. Currently, it consists mostly of well-established in vitro methods, those that have already been through the OECD validation processes, the ECVAM, the ACVAM, a lot of well-established QSAR models and so forth. And then a corollary to that is that we have under development basically a process by which many of these new and emerging NAMs then could get placed on that list. So we have yet to articulate just exactly what that process is, but it is underway. And then moving to some examples from our pesticides program. Again, I mentioned that they have actually been on this path, having a very animal testing intensive data requirements associated with registering a pesticide. For one thing, that gives them a lot of data to look back upon and figure out strategically where one might go about making reductions, finding those places where maybe the requisite data is not particularly useful or has not been historically used very often and so forth. So they've been very strategic and have been at this for some time, putting out a strategic vision. for adopting new approach methods some time ago. That the original vision, I think, goes back to like 2013 or so, and you can read about it on the webpage. There's also very specific guidance out there around waivers. So again, as I mentioned, FIPRA has a long list of testing that is required to come into the agency with a pesticide registration package. But through evaluation of existing data and the emergence of NAMS data, the pesticide office has come up with guidance on certain tests that could be waived. So you can waive out of the standing requirements if certain conditions and alternative methods are provided. Also, they are well on a path to outlining strategies for replacement of specific tests with NAMS, and we'll get into some of those momentarily. Another area in the pesticides program, there is a requirement for a bunch of acute testing to be done. Certainly as part of the active ingredient, but they also have something called the 6-pack, which is a list of required tests for any pesticide formulation. So in a collaboration between EPA and Nicole's group over there at NIEHS, NICEATM, again, I mentioned that the pesticide program has accrued a lot of test data over the years. So they analyzed the relative contribution of the data from acute oral and acute dermal tests to what ultimately goes on the pesticide label, which is essentially classification and labeling exercise. So they looked at the pesticide formulations in 2016 and active ingredients in 2020. And so this is what underlies this guidance that's out there now about waiving some of these tests in light of the look back on their utility from gathering all that data, and also because there are emerging alternative methods coming online. Uh-oh. Having a little mouse problem here. Sorry about that. And furthermore, on this acute oral pesticide toxicity testing, you saw, this is a little bit of a repeat, but also a shout out, this Catmos, this collaborative acute toxicity modeling suite that Nicole can speak to much better than I, but nonetheless, using that data and doing some analyses which have been published, as you can see there, this modeling suite is developed, and there's a product of a workshop. Basically, it is an in silico method that predicts acute oral LD50s based on the structure. So a computational approach that could be used in lieu of animal in vivo testing or oral tests. Another area that the pesticide and toxics program have embraced the use of new approach methods within for meeting the regulatory requirements or conducting our chemical risk assessments is in the area of eye irritation. This, I think, Amy's paper, Clippinger et al. Was cited in Nicole's talk too. But that underlies basically a testing framework for assessing eye irritation potential, which is a requirement under the pesticides regulations. And very specifically for antimicrobial cleaning products, there are these 3 NAMS assays. So they're either in vitro or ex vivo assays that can be used in conjunction with one another to meet some of these requirements for classification and labeling. So there, you can read more there at that website. And it is something that is currently being considered on a case-by-case basis in the pesticide program and in the toxics program, because we don't have mandated upfront testing requirements. Certainly this type of data that would come into us under TSCA for a new chemicals analysis would also be much appreciated and considered, given our mandate under TSCA to move in this direction to reduce animal testing. And just kind of a testament to the uptake. Again, some of these methods have been, we've accepted them at EPA, at least for consideration, looking at the pesticide program, doing the waivers. But in the area of skin sensitization, another example here is that as far back as 2018, we had a draft interim science policy to use a collection of NAMs, an alternative approach to looking at skin sensitization. And that was put out there, and basically EPA saying we will accept these, each of the assays themselves were already validated through the OECD, but as a collection, they ultimately became a defined approach. But EPA was taking in and accepting these data so that we could become familiar with the methods, but also kind of, build the confidence among our staff, but also the greater community that in fact, these in vitro NAMs methods can serve the purpose of the previous animal tests within the regulatory context. So both the Pesticides Program and the Toxics Program have been accepting the assays within the defined approach. I may as well go to the next one, which eventually just this past year became a test guideline in the OECD. So a little bit of the point here is that building confidence, all of the elements in that confidence framework and much of the validation work that Nicole talked about all needs to happen, but actually trying to evaluate and consider these methods that are within the development process can be synergistic to building confidence. So start to consider them earlier within the regulatory process, even before something might become completely signed, sealed, and delivered, if you will, as a test guideline. So certainly we have a lot of flexibility in the TSCA world for that. Pesticides has, again, because of their statutory requirements, a little bit more confinement, but clearly they've been moving forward. And all of us use the principles of best available science to consider these emerging methods within our regulatory contexts. Some other work that's going on, again, is to take that skin sensitive sensitization in vitro methods. You heard from Nicole about, and some words from me earlier about domain of applicability. Certain tests fit and are very suitable for certain chemicals. And certainly when something's in development phase, usually you have a certain test set of chemicals that are part of the validation process, but then the expansion of these NAMs methods to a greater domain of applicability as far as chemical space is often a necessary next step. And it shouldn't have to all happen at once, so long as we're very transparent about describing the domain of applicability at any given time. But there's work going on here, for example, to test additional chemicals and evaluate whether these methods can be, the domain of applicability of these methods can be expanded to more and more chemicals. But certainly it can be done in a stepwise fashion, so long as we're transparent about where you are at any given time. So just to conclude, hopefully, and you saw from Nicole, and I tried to acknowledge as well that this progress towards the uptake of NAMs, especially in a regulatory context, takes collaboration across many, many sectors. And I think that this NAMs arena is one that is very much on the forefront of that kind of collaboration. It's broad and wide, and a lot of excellent science coming out of it. Certainly we need to be transparent. Both Nicole and I stressed that, and we need the peer review to occur with any given method or group of methods or IATA, whatever it is, we need to vet it. But also, hopefully, some of these examples provided as to what some EPA program offices are doing demonstrate that learning by doing is also another component that we can apply these fit for purpose methods to address real world issues. And we are doing so at the EPA in our chemical programs. With that, I'll thank you very much and acknowledge a lot of people that have been working in specifically on the competence framework here at EPA.
Okay, well, thank you very much, Tala. That was a great presentation, really informative. And I think it tees up a lot of questions for the discussion. So folks, we have a couple of clarifying questions. Keep your questions coming in. I'm gonna bring these to both of our speakers right now, but most of them I think are gonna be good for the entire panel. So I'll save most of them for the panel. So with that, we have a specific question for you, Nicole, about the updated ECVAM validation guidance, asking when it's expected to be published.
Great, thanks for the question. So I don't know is the answer. We are hoping to have it like a final draft for agency clearance in the spring, maybe even a little earlier. I don't know, like early spring, you know, but as you can imagine, getting clearance from 17 different agencies takes a minute. So, you know, optimistically, I would say, you know, by the like late spring, early summer of next year, we might have a draft out for public comment. And it won't be finalized until we get public comment, you know, at least have one round of public comment on it, probably have our SACATMs, our Scientific Advisory Committee on Alternative Test Methods, comment on it, and then hopefully, you know, finalize it by the end of next year.
Okay, thank you. This question came in during your presentation, Tala. It asks, to what defined approach does key event 3.1 STS refer to? So they understand what key event 1 and 3 are, but maybe elaborate a little bit on the approach.
Okay, Gina, you might have to help me here. Basically, this is about the defined approach for skin sensitization. It has to do with there are 3 different, I think there's actually more now, but there are 3 different in vitro tests arrayed in a scheme, kind of a decision contact scheme. And there's Nicole or Gina are gonna have to get into the absolute details, but you have to have 2 of the 3 things signaling one way.
Can I jump in, Tala? Because
yes,
what you're describing, yeah, what you're describing is the 2 out of 3,
right?
Which is one of the DAs under the science policy. The other one, the key event 3/1, is a sequential testing strategy, and so that's one where you test key event 3, and so that is you know, the first was the HCLAT, but now there are a number of test methods that are validated under OECD TG 442E. And so the science policy, and this is an important distinction, the science policy indicates that any of the methods that are included in that key event-based test guideline 442E could be used for that key event 3 method. And so then if you have a positive in that, then you're done. It's, you know, it's classified as a sensitizer. If you have a negative, then you go to the key event 1 based test method. And again, the original method that was validated under 442C was the DPRA, but the direct peptide reactivity assay. And now there are other methods that are included in 442C. So again, it's basically any method in 442E as key event 3 as your first test, and then sequentially, if you get a negative, going to any method in TG 442C as your second test to cover that key event 1.
Thank you, Nicole. She obviously is one of the developers here.
Yeah.
But can you say, Nicole, is it all of that decision tree network described in the guideline 497?
So that particular method is not part of the DAS guideline 497. It predated
Afterward.
guideline 497 actually. So it was part of a large international collaboration between Cosmetics Europe and NICEATM to evaluate a number of different defined approaches, and it was found to be every bit as robust and reliable in predicting the human hazard endpoint for skin sensitization. And so based on those publications and all of the peer review that was associated with that project, the EPA was actually several years ahead of the OECD in adopting those defined approaches where there had been a lot of confidence demonstrated in their ability to predict hazard.
So
And
where could we send somebody to read up more specifically about it in the interim policy?
It's described in the policy, in the draft science policy that EPA put out. And then there are
on my slides, I didn't put a link to that one. But I think if you just Googled,
I think we can grab a link and put it in the chat.
Yeah. And then if they want additional details, the publications that it's based on are cited within that policy.
Yeah. Great. Thanks so much.
So one last question that came in that I thought one of you could address, and I'll let either of you jump in. And they're asking about the concept of applicability domain with IV-IVE approaches to derive estimated doses in humans. Just in general, anyone want to tackle that? So in other words, how to use the concept of applicability domain for these IV-IVE approaches, or maybe what are the main concerns or considerations?
I mean, I can always talk, but you guys have heard a lot from me. So João, Warren, do either of you want to weigh in on that?
Well, maybe we'll just...
Yeah, I think João was answering. He was muted though.
No, I wasn't answering, but I unmuted myself. I don't see why. I mean, in terms of in vivo extrapolation, applicability domain. I don't understand really what the issue would be there, to be honest. Not sure exactly what the question wants.
No, I was thinking, I wonder if there are particular issues with some of the models that are like.
So
you have to understand, at least from the in vitro part, you know, what happens to the substance you're testing, right? But there are models for that, you know, understanding through internal exposure to the cells, what's binding to plastic, what, you know, the fate of the substance, to understand exactly the doses that the cells are seeing. I think that's extremely important. And in that part, of course, applicability domain is important, or at least understanding the characteristics of the substance you're testing. So from that perspective, yes, it's important. The same for in vivo doses. Understanding internal doses would be important in order to have proper comparisons being made. But beyond that, I don't know, Nicole, if you want to add something. You were just going to answer.
Yeah, so I mean, my own interpretation of this question has to do with things like we only have experimental values for some of the important like ADME and PK relevant parameters for a limited number of chemicals, right? So we have information on intrinsic clearance and plasma protein binding for anywhere from several hundred to several thousand chemicals, but we have, built pretty reliable, depending on the endpoint, QSAR models based on those training sets to predict those key parameters that would allow you then to do IVIVE for a much broader range of chemical space. And so, for example, OPERA, the open QSAR suite of models that's led by Kamel Mansouri at NICEATM and in collaboration with EPA, that has models for predicting, intrinsic hepatic clearance and plasma protein binding and tissue partition coefficient parameters and things like that. But there are applicability domains associated with those models, and there are confidence, metrics associated with those predictions. And so OPERA provides that information to the user. But then also from a broader perspective, the applicability domain of being able to use in silico parameter predictions to do IVIVE is limited to compounds with defined structures, or organic compounds that can be run through those QSAR models. And so, being upfront about the sort of limitations of that type of an approach when it comes to UVCBs or complex mixtures and substances where you don't have a defined structure, I think is important as well.
Yeah, thank you both. And I think I'll go ahead and introduce our other panelists since they're here in front of you now. So many of you probably already know them anyway, but our panelists are also both experienced in the evaluation of toxicological test methods. They both have had impactful careers in this space and have been involved in many of the validation approaches that are currently available. So Dr. Warren Casey, you can see on your screen, he's a senior science advisor in the Office of the Director of the Division of Translational Toxicology at the National Institute for Environmental Health Sciences. He has serves as the executive director in the U.S. Interagency Coordinating Committee on the Validation of Alternative Methods, or ECVAM, and previously directed NICEATM. Prior to joining NIEHS, he worked at GlaxoSmithKline for about 15 years, and he has also co-chaired the OECD Validation and Management Group, non-animal, and serves as NIEHS Principal Representative to the Tox21 Interagency Consortium. Thanks for being here, Warren.
Thanks very much for having me, and thanks for the partners for putting all this together. And I just quickly want to say that, just seeing that the partners decided to do this together really typifies why I think we've made so much progress in the field of NAMs. I mean, look at everybody here, the speakers, the panelists, the organizers, all work together so well, and that has not been the case in the distant past. And that collaboration should give everybody a lot of hope that we're making a lot of progress, significant progress, and NAMs are going to be incorporated into chemical safety assessment in the near future. So thank you.
I'll take that prediction. Okay, so also joining us is Dr. João Barroso, who's the scientific officer at the European Union Reference Laboratory for Alternatives to Animal Testing, or EURL ECVAM. I always struggle with the first part of that, which is part of the European Commission Joint Research Center. João coordinates and conducts scientific and policy support activities in the areas of alternatives to animal testing, including validation and peer review of non-animal methods and approaches, and their translation into EU legislation and international standards. Thanks for joining, Joe.
Thanks, Christy, for the kind introduction, and thank you so much for inviting me to be in this panel today. It's always a pleasure to discuss frameworks on building confidence. I think validation is an evergreen concept. It remains important, but it needs to evolve as science evolves, and I think that's what Nicole and Tala were presenting today. And I think it's great that we're trying to again find ways to make it more efficient, to make it more valuable, because we really cannot live without it actually for when it comes to regulatory toxicology and regulatory sciences. So happy to be here today, and I agree with Warren. I think the future looks bright. And yeah, we need more positiveness. It's not just about frameworks, it's about mindsets as well. And I think that's the most important thing, changing mindsets. Thanks.
Thanks, Cheryl. Okay, so maybe continuing the discussion that we had on applicability domain, there have been a couple of questions coming in. One of them is maybe the suggestion around how we think about applicability domain. So someone asks, does each NAM have to be tested with each type of substance or product to demonstrate applicability? Or can it be assumed that the NAM is applicable unless there is information to suggest otherwise? And then another question relates similarly to specifically to testing mixtures. And wondered what are some of the challenges for expanding the applicability of certain kinds of NAMs to mixtures. And maybe when you're responding, think about the differences between maybe in vitro and in silico models and how we think about applicability domain there. So panelists, please go ahead.
Maybe I can start with the first part of the question. I think we should probably not go into the error of separating chemicals according to sectors, because chemicals are chemicals. Actually, several chemicals are used across sectors for different utilities. So I think we need to actually think much more about the concept of chemical space, understand the chemical space that a method is able to cover, independently of how that chemical space is used then by the different industrial sectors. Because, you know, requesting specific validations for specific substances or materials that are used by specific sectors is not very efficient. Moreover, when we're thinking about mechanistic methods, I think what is important is to understand whether the method, mechanistically speaking, is working well. And that you can understand with just a few chemicals that actually acquire that mechanism of action. And it's less important whether a chemical is a cosmetic or a pharmaceutical or a pesticide. But again, we need to understand the chemistries. We need to understand what chemical space those types of chemistries cover. And there are robust methods to look into that and to characterize that, that actually consider multiple factors and just make sure that you are covering that well. And if you do that, then it's less important than what type of sector you're trying to address. Just then for the mixtures, honestly, I think I don't really see why mixtures would not be applicable to in vitro methods unless there are certain technical difficulties that do not allow the testing. But you know, in the OECD so far, usually methods are considered applicable to mixtures unless there is information showing that they don't. And then we need to understand also when they don't, why are they not working? And most often, the conclusion that they're not working, it's because they're not concordant with in vivo data. But we also know that many times in vivo data are not completely reliable. And I've seen examples where the concordance to animal data is not that great, but when you have actually human data, they are much more concordant with the human data. So, you know, I again, as I said, I think I wouldn't see any limitations to mixtures. And if there should be some mechanistic explanation to that, or a technical limitation of the method, where some of the components of the mixture are not soluble, or you know, especially for water-based methods. But for the rest, I don't see a limitation. I don't know if the other colleagues in the panel want to add something.
I would add, so I agree with what you said. I think we need to focus on the chemical space, but we also need to make sure that the end users, with whatever agency, is comfortable with the chemical space covered. And it's not the test method developer picks 30 chemicals that they know are going to work and then expect somebody to take. So I think that partnership, that the agencies have confidence that even though there may not be, that their chemical space is covered, even though it may not be distinct from others, as long as theirs is covered, I think we need to and what you said about the mechanism is spot on. If we don't understand the mechanism, if you're looking for an estrogenicity assay, you could have that direct binding, or it could be a secondary or tertiary response. So we need to understand the limitations of the mechanism that we're evaluating as well.
Anyone else want to jump in? Okay. There was another question about gaining confidence and about reference chemicals and controls, and whether it would be envisioned that these would be endpoint and route of administration specific. And this kind of hits into this applicability domain conversation. And I know there's a lot of conversations going on about how to make validation more flexible and amenable to different types of NAMs and their context of use. And so I wonder if anyone has any comments about what your kind of vision for how reference chemicals and controls can be chosen or should be determined.
So I mean, I can start us off. I think reference chemicals is it's a really hard and really important question, right? Because that's kind of the crux of being able to demonstrate performance of a new method, is to have something to compare it to. And that something, is by and large going to be reference chemicals. I think we're encouraging also incorporation of, what is the biology that's being measured, because I think the reality of the situation is that there are some endpoints, a very limited number of endpoints for which we have a lot of reference chemicals. So, estrogenicity, anti-androgenicity, so some of the endocrine disruption endpoints, the acute topical toxicity endpoints, the acute systemic tox endpoints, those types of endpoints, we have good reference chemicals. But the more sort of multifactorial the disease becomes, the more complex the biology that's being represented, the more challenging it is to not only identify a list of reference chemicals, but understand sort of at what level should those reference chemicals be delineated. Like, do you want reference chemicals that are just like carcinogens versus non-carcinogens, or is that like way too far down the path of biology in terms of trying to predict an apical endpoint? If we're trying to step it back and take a more mechanistically informed approach, can we break down the biology into more discrete pieces and say, okay, here are some reference chemicals that we know cause, excess, like chronic inflammation. Here are some reference chemicals for genotoxicity or mutagenicity. And so breaking it down by mechanisms and developing reference chemicals at the level of biology that we're actually trying to start predicting, I think is the direction that we need to go. And there's some work being done there. The problem is that a lot of our existing reference methods, so those animal methods, aren't going to give us that information to be able to derive those reference chemicals. And so it becomes a little bit of a circular argument, but I think once you start to bring in enough data and you start to look at sort of consistency of response among different assays and different types of technologies that are trying to measure the same biological process, and you start to see subsets of chemicals that are consistently causing oxidative stress or consistently upregulating inflammatory cytokines in multiple different test systems, then you can start to build that sort of weight of evidence and that confidence that these could be reference chemicals for new methods that are trying to measure that same biology.
Yeah, I agree with everything Nicole said, and just sort of a simpler way of saying it is that to the question that Annie posed, it could be, it depends to me when I think about it, is what is the new approach method trying to measure? And if it's you know, a lot of these in vitro things, they take all different types of methodological formats. So depending on what it is you're trying to measure, some chemicals might be amenable to that test system and others not. So you might have to, and it gets to Nicole's point, if you're measuring pieces or parts or events in an overall AOP, you just have to consider what the test system itself can do. And, you know, I think Nicole was saying, if you have this mechanistic pathway, you might have different reference chemicals for different pieces along the way. But that's why, you know, the transparency and description of the method. And it, again, one of the things I was trying to stress, I hope it came through, is that you can validate a method with a narrow applicability domain at first and use it, knowing with that knowledge, use it properly, while work continues to go on to expand the chemical domain. And maybe you have to tweak the system or use another alternative method for certain other chemicals, but still fits within the same adverse outcome pathway. So it's not like you have to build it all to use part in a progressive way.
Yeah, thank you, Tala. Any other panelists want to jump in?
Actually, just to add to this topic and the previous one we were discussing, which are kind of related, I saw something, a clarification coming in, saying that the question about substances, substance types was referring to chemical classes rather than industrial sectors. And since I answered that in the beginning, I would just add to this bit in terms of chemical classes, that my experience is that it's very difficult to make such an analysis. First of all, because most chemicals belong to much more than one class. Chemicals today are not just, you know, belonging to a single class. When you do an analysis by, for instance, organic functional groups, you'll see that one single substance will cover many. Then in terms of validation, you end up having just a few out of each of those organic functional groups, and it's quite often very difficult to make assumptions on applicability domain on the basis of 2 or 3 substances for a specific class. And sometimes you may actually end up having wrong conclusions. When, you know, I've seen examples where a certain conclusion was devised based on an analysis of applicability domain on the basis of chemical classes or organic functional groups. And then when you actually do other types of analyses, you actually find a specific mechanistic limitation that could explain why a certain substance was not detected properly, which has nothing to do with the chemical class, for instance. So as I said, you know, you can actually do a much more robust analysis of chemical space, which includes also chemical class and includes other characteristics, which probably is more informative than just looking at organic functional groups or trying to distinguish chemicals according to specific classes. That's something we did in the past, but I honestly don't think that's quite often very informative when you have lists of validation studies that are done on the basis of 20, 50 chemicals at max, you know, and you cannot cover all chemical classes in a statistical significant way to actually make conclusions out of that. Other analyses are much more robust, I think. I don't know if the others want to add something to this, but, you know, just wanted to cover that bit there.
No, I agree. When we start classifying, saying alcohols are a chemical class, I mean, that's an enormous space. So yeah, we have to get better about how we define that chemical space, I think.
Great. So we are really at the end of our time. I don't know how that happened so fast. I'm sorry that we didn't get to all of your questions. We really appreciate your participation, and we've gotten a lot of compliments in the chat for our speakers and our panelists, so we appreciate your time very much. We appreciate your input, and I just want to encourage you to look out for our next session, session 2, where some of our panel speakers today will be joining us, and we'll have some new speakers, so we'll be able to continue the discussion. Thanks again, and have a great day.
Thanks, everyone.
Thanks, everybody.
Thank you.